So you're focusing on those two, or you're focusing on studying biology?
On also I have human bio on the same day.
On the same day? Oh
Yeah.
my. Well, there are some common topics, but you have the skeletal system and you have the ear and you have different topics in human biology. This is so unlucky to have them both on the same day. Well, best of luck. Let's solve this exam. We did not solve this exam, so we don't waste our time. You can all see my screen?
No.
Okay, 1 minute. Now, can you see my screen? Hello?
Yes, Miss Vacancy.
Okay, we did not solve this exam. Let's solve it together. Fine, I'll go straight to the questions, but of course, I keep on saying you don't do this on the exam. Read the question from the first word. You have a picture here, and they're asking you to identify the stages of mitosis occurring in the cells labeled B and C. So let's go straight to B. What will you say about B? Which stage of mitosis is this?
Prophase?
Excuse me?
Prophase?
Yes, it's prophase. Now, this question is easy and tricky at the same time because they said identify the stages of mitosis. If they did not say, if they said identify the stages of the cell cycle, then you might be confused in B and cell and tell me that B is interphase. But is interphase part of mitosis?
No.
No.
No, interphase is not part of mitosis. Mitosis is prophase, metaphase, anaphase, and telophase. So we cannot say interphase. So B has to be prophase. In prophase, the chromosomes are condensing. This happens in early prophase. So B is prophase. Now, what is C? Also, C is a little bit confusing. What is C? You can see that the chromosomes started to arrange themselves in a certain place inside the cell. So what is C?
Metaphase.
Metaphase.
Yes, C is metaphase. Okay, so this is easy. So B is prophase and C is metaphase. Describe the behavior of the chromosomes in the state of mitosis shown in cell A. Let's go to cell A. So cell A, let's identify it and then describe it. And we want to describe it in 3 points because it's 3 marks. So what is A?
Anaphase?
Anaphase.
Yes, it's anaphase. You will take 1 mark for that, but still we want 3 descriptions. How can you describe A?
The centromere split and the
Yes.
chromatids start moving to opposite poles of the cell.
Yes. What else?
The spindle
So A?
fiber shortening.
Yes. Okay. A is anaphase, and we want 3 descriptions because I have 3 marks. Okay. If you want to have, or if you have 4, go ahead. So let's say one of them, the centromeres, they will split or they will break. And the sister chromatids, they will, what did you say, Farouk?
Move to the
They
opposite
will
sides of the
separate.
opposite
Yes.
poles.
And they will move to opposite poles. Yes. Move to opposite poles. Now, what else will you say? Give me a third point.
The spindle fibers are shortening.
Yes, spindle fibers, they will contract or they will shorten or whatever, pulling the sister chromatids to opposite poles. Good. Now, when you come to draw anaphase, because many times they ask you to draw anaphase, draw a cell with the centrioles, 2 centrioles, each at a different pole, and the spindle fibers are shorter, and the sister chromatids, they look like this. The centromeres are pulled first, and then the 2 arms are like extended forward. Okay, you have this picture. So, and let me magnify that. This is anaphase. Most of the time, they ask you to draw anaphase. The spindle fibers are shorter, they will contract, and the sister chromatids, when they are broken at the centromere and they're being pulled, they look like this. They're pulled backwards. Okay, centromeres first, and then the arms are extending forward. And of course, you have another group on the opposite poles. Describe the role of the DNA ligase in interphase. This is easy. What will the DNA ligase do? I want three keywords.
In the semi-conservative, in the synthesis phase, in the semi-conservative replication of the DNA, it joins the Okazaki fragments in the lagging strand.
Okay, this is not enough. Yes, it is used in the replication of DNA and it will join the Okazaki fragments. What exactly does it do while it is joining the Okazaki fragments?
It
And
forms
yes, this
phosphodiester
is the
bonds.
lagging. Yes. And yes, this happens in the lagging strand only. Yes. What does it do? It will form the phosphodiester bonds. Cool. Or the sugar phosphate backbone. I cannot write things properly on the screens. This is what I always want you to take care of. Before you answer the question, look at the number of marks. 3 marks, then minimum 3 sentences. Minimum 3. You can write 4. So if you tell me it will be used in replication, DNA, semi-conservative replication, it will join the Okazaki fragments. In the lagging strand, you might take 3 out of 3 or 2 out of 3. Why? Because maybe they want that particular fact. That what do they do exactly? They form the phosphodiester bonds. I'm not going to look at the my scheme to not waste time, but I'm sure that they will mention something about the phosphodiester bonds between the Okazaki fragments. So look at the number of marks so you have a guide and know how much you will write. And it's always more is better than less. So writing more is always better than writing less. Okay? State the location of the telomeres on a chromosome. This is easy. Where are the telomeres found?
On the ends of the chromosome.
Of course. State means one word on the end of the chromosome. Some people with a long lifespan have cells showing a higher than normal activity of telomerase. You definitely know what it does. Suggest why a long lifespan could result from a higher telomerase activity. What is the function of the telomerase activity?
It repairs the telomeres, or
It's what?
it repairs the telomeres or keeps the telomeres alive for longer.
I cannot hear you properly, Farouk. Put up your voice.
It repairs the telomeres or like keeps the telomeres alive for longer, or it means
Yes, correct. After every replication, the telomeres, they normally shorten. When there is a higher telomerase activity, the telomeres will not shorten. The function of the telomerase, this enzyme, is that it keeps on adding telomeres at the end of the chromosomes after every replication so that the length of the DNA is maintained, so that the genes are not lost. And this will help with more and more DNA replication. This will help with more and more mitosis. This will prevent apoptosis. Okay, go back to your notes and study the function of the telomerase activity and remember when it is active. Normally, when the DNA is replicated, the telomere will become shorter and shorter and shorter until it is finished. And when the telomere is finished, the telomere is found at the end of the chromosome. It's here. Okay, when it is finished, the DNA will not be able to replicate and the cell will die. This is normal cell death. This is apoptosis. Now, the telomerase, the enzyme, keeps on adding on the telomeres after every replication to prevent. So you will write the function of the telomerase activity. It keeps on adding to the telomeres after every DNA replication to prevent shortening of the DNA, to prevent loss of genes. And this will help in more DNA replication or less DNA replication.
more.
Yes, so this will help in more DNA replication. In other words, this will help in more mitosis. This will prevent programmed cell death or apoptosis. And this enzyme is active in 2 situations. Give me 2 situations where this enzyme is active. Normally, when is it active?
In cancer cells,
Yes, but this is not a normal situation. This is an abnormal situation. But yes, they are
After cell
active in
replication.
cancer cells. They are active when you are an embryo in the stem cells, when you were still a fetus, because the fetus has a lot of growing to do. Okay, so don't forget. So the telomerase, they are highly active when you are an embryo in embryonic cells and in cancer cells. And the telomerase activity is an indicator of whether you have cancer or not, God forbid, because they should not be very active in adult cells. But when they are, this means that this adult cell is rapidly dividing uncontrollably and it is cancerous. Don't forget this part. Okay, describe the trends shown in figure 2.1 between 1980 and 2000. This question does not depend on how much you studied. It depends on how well you can read a graph. Now, when you are asked a question like this, I highly advise you that you break it down so that you do not miss anything. When they tell you describe a trend between 2 years, then you will write some information over this year and some information over this year. So you can organize your answer like this, especially those that have very horrible handwriting and they have very messy answers. Try to at least organize the answer. Your handwriting, this is something you cannot control, but at least organize the answer. So you can say 1980, you will discuss, and then 2000, you will discuss. Okay? Describe the trends. You will just describe the graph. You will not explain anything. Now, I will give you general information describing any graph. This is so easy. The graph is going down, then it is decreasing. The graph is going up, it is increasing. Here is always a peak of anything, peak or maximum. Okay? And if you have two graphs, let's revise everything. If you have two graphs, and they meet at a certain point, then this is a meeting point, or they both meet at a certain value. Okay? And if you have a graph that is going up and down like this, how do you describe it? Hmm. You can say up and down, or you can use a better word, fluctuate. Fluctuate, yes. And when the graph is moving upwards or downwards, whatever, and then it will reach a constant. This is very easy. It will become constant, reach a plateau, it will level off. They like those terms. Now here, very easy. You will go to 1980. 1980 is here. The number of reported cases was nearly maximum. Give a number, write the number down so that you take the grade. You don't just say that in 1980, the number of reported cases of TB was very high, because this is what you write for me. You will deduct a mark. You will lose a mark. Why? Doctor, I said it's very high, but you did not give me a number. So in 1980, it was very high. It was 28,000. Give me a number from the graph. They are here examining your mathematical skills. They want to know if you can read a graph properly or not. At 2000, what happened? There was a great drop, obviously, in the number of reported cases of TB, and they reached, and write a number, they reached nearly 16,000 or 16 and a little bit. So you have to describe. Okay? There was a decrease. You can even subtract and know how much there was a decrease. This is called a trend. Increased, decreased, fluctuated, whatever. Okay? So describe the trends between. So you will not only describe what happened at this year and at this year. No, you will describe what happened throughout the whole graph. It was high, it was 28,000, and then it decreased until 1985. It dropped till nearly 22,000. It increased a little bit. It fluctuated a little bit, and then it increased again and reached its peak at 1992 or 3. You will have to find out for yourself. And then it decreased again until year 2000 to reach its lowest levels, which was around 16,000. So you will break down the graph bit by bit in order to take the 4 marks. Okay, so here they did not ask you specifically what happened at those 2 years, but in between. So all the years, actually, you can break it down to all the years. Am I clear? Explain how an increase in the number of people infected with HIV could have affected the number of TB cases. Now, without even looking at the graph, the more the HIV, the more or the less the TB.
More.
Of course. So you will have more HIV, more TB. Now, why? Because they said explain.
HIV causes a decrease in the T lymphocytes, therefore
Perfect.
TB becomes an opportunistic infection.
Perfect. This is enough for 2 marks. HIV, of course, will drastically destroy your immune system. It directly affects the number of T lymphocytes, they decrease, and any opportunistic infection, the word opportunistic is important, such as TB, you can easily be infected with opportunistic infections because you have no immunity. That's it. Very easy. This is an O-level question. State two ways that the impact of antibiotic resistance can be reduced. Again, this is an O-level question. How can you prevent antibiotic resistance?
Taking the full dose.
Excuse me? Taking, I think you said proper dose, Mariam?
Yes.
Yes. You will take the proper dose. What else? This is O-level also.
Complete the course.
Very good. Complete the course. Now, yes, they are only two, but what else?
Not taking antibiotics for viral infections.
Of course, don't take it when you have a viral infection. Don't take it frequently. Don't take it when it is not necessary. Use a different antibiotic every time. Combination of antibiotics. What else? What else? Proper isolation of an infected patient. Proper hygiene. Any one of those. Now, the cartilage in the bronchi can be damaged, and some people with TB suggest the effect that damage to cartilage may have on the bronchi. It's so easy. If there is no cartilage, what will happen to the bronchi?
It will collapse.
Again, what's this? It's a very easy exam. Of course, it will collapse. It will close, blocking of the airways, no support. Okay, now this question also. We want to know the cell type and where is it from. So the ciliated cell, the ciliated epithelium cell, is it in the alveoli or bronchus or trachea?
Bronchus and trachea.
Of course. Everything actually is in the bronchus and the trachea. This is the bronchus, not the bronchiole. So everything is in the bronchus. Nothing, no cilia in the alveoli. Goblet cell,
Bronchus and trachea.
yes. And no alveolus. Smooth muscle cell,
Bronchus and trachea.
yes. The alveoli, don't forget, is just a simple layer of cuboidal cells. That's it, surrounded by many capillaries, and we have loss of elastic fibers. No cilia, no goblet cells, no cartilage, nothing. So the smooth muscle cell, again, squamous epithelial cell, squamous, not ciliated.
الالفيولاي بس مش في البرونكس ولا التركية.
Excellent. Now this seems like an easy exam. Let's see. Identify X and Y. This is O level also. What is X?
neutrophil.
Yes. And what is Y?
monocyte.
Yes, lymphocyte, monocyte, both correct. Okay. Now, this is write down the formula you will use to make your calculation. It's definitely magnification. Yes, what's the, come on, quickly. What's the formula for magnification?
Image over actual.
Yes, I am image over the actual times the magnification. Okay. They want to know the magnification. So the magnification will be the image over the actual. Describe how carbonic anhydrase is involved in the release of oxygen from red blood cells at respiring tissues. This is a very nice and direct question. Can someone tell me what happens or what does the carbonic anhydrase do?
It catalyzes the reaction between water and carbon dioxide, forming carbonic acid. Carbonic acid dissociates into hydrogen carbonate and hydrogen ions. So the red blood cells have a higher affinity to the hydrogen ions, so they release the oxygen and bind to the hydrogen.
Perfect. What you just said is summarized in the equation, but of course you will write it down. Now, here, I don't want you to do this. Hydrogen plus hemoglobin will give me hemoglobinic acid, which is correct. Why? Why? Because it's better to write it like this. The hydrogen plus C hemoglobin, excuse me, will give me what? Who will tell me? Here, oxygen will dissociate. Excuse me, what did you say, Mariam?
It will give us hemoglobinic acid and oxygen.
Yes. This is the better reaction. They're both correct, but the hemoglobin is always bound to something. The hemoglobin is never free. The hemoglobin is very busy. It is either bound to oxygen or hydrogen ions, depending on which has a higher concentration. Oxygen is more, so it has a higher affinity to oxygen and it will bind to oxygen. hydrogen is more, then it has a higher affinity to hydrogen ions and it will bind with hydrogen. So, okay, what Noor said is perfect. Good. And you can summarize
Yes, it
it. You can
was
write what he said. Huh?
فاروق مش أنا.
It was who? Farouk? Oh, Farouk or
فاروق
who?
yeah.
Adham. Like, you're all the same to me. You're all amazing. Good. So what you said was perfect, and you can supplement that with the equations. It would be amazing. Here, sketch a curve on the figure to show the effect of the sulfonamide on the rate of reaction catalyzed by carbonic anhydrase. I have to read the question to see. Yes, it's a competitive inhibitor. Who will remind me what is the graph for the competitive inhibitor?
It reaches the same Vmax but has a higher Michaelis constant. So
Very
it will
good.
be under it, but it's still reach the Vmax.
Okay, perfect. Don't forget the competitive inhibitor and the non-competitive inhibitor. Both of them, they reduce the initial rate of their action. So it has to be below it. Okay. The competitive inhibitor affects the affinity. It lowers the affinity, but the non-competitive inhibitor, does it affect the affinity? Who remembers? We're just revising both together now. The
No, it does not.
non-competitive, it doesn't. Yes. The competitive inhibitor will eventually reach the same Vmax because with time, an increase in the substrate concentration will cancel out the effect of the competitive inhibitor. Good. I can see that you all improved. You all studied a little bit more in the past days. Now, here, complete the table by writing the level of protein structure that applies to each description. Let's read it. A gliadin protein is a single polypeptide that forms a compact structure. This is which protein level?
Tertiary.
Very good. Why did we say tertiary? Not primary. I was expecting you to tell me primary because it said here single polypeptide, but it's wrong. It's not primary.
Compact
It's tertiary.
structure.
Very good. We had a single polypeptide chain that started folding to give me a compact structure. Very good. 20% of the amino acids in a glutenine molecule are glycine. So here they're talking about the amino acids. So this is what?
Primary.
Primary. Very good. Okay. The gliadin and the glutenine molecules contain regions of the beta fleeted sheets.
Secondary.
Secondary.
Second. Okay, if you have an easy exam like this, number one, you need to worry because an easy exam, this means that the curve will be very high. So when you have an easy exam, your answers should be perfect. Perfect, because the curve will be really high. A difficult exam, of course, is not nice, but at least the curve is lower. Okay, so this is a very easy exam. Explain how a lack of introns in a gliadin gene affects the production of messenger RNA from the primary transcript. There are no introns, only exons. So what will happen? So easy. Mm.
The exons are fused together and they form a polypeptide chain.
The exons are fused together. The exons are already fused.
A polypeptide chain is formed during the translation.
Yes, in all cases, the polypeptide chain is formed during the translation, but there's a step that was done to remove the introns. Now we don't have introns. What was the name of this step?
Splicing?
Splicing.
Yes, splicing. Now, do we have splicing? Will this step take place?
No.
No.
No, no splicing. What you said, Maryam, unfortunately, it's not going to be correct because in all cases, translation will happen, and in all cases, the exons will be converted to a polypeptide chain. But what normally happens is that we will take a copy of the DNA and we will create a primary transcript. This primary transcript or primary RNA contains both the exons and the introns. Okay. And then there is a post-transcriptional modification where we will remove the introns because they are non-coding and we will fuse the exons together and then we will translate. But now they're telling you that there are no introns. So there is no need to remove the introns. They're not there anyway. So there will be no splicing. The exons will go immediately to the ribosome and they will be translated. Do you understand me? Now we need to look at the mark scheme for this question. 1 minute. I have the mic scheme here. This is number what? 4 AII. Okay, let's look. Okay. Any one relevant example, RNA or gene splicing is not needed. Yes, it does not occur. Fine. All the primary transcript is a coding sequence. All the primary transcript, like we said, it will be introns. They will be, it will be exons. There will be no introns, like they said. Okay, so no nucleotides removed from the primary transcript. No splicing. No introns will be removed because there are no introns anyway. Good. So this question is easy, but again, the question format itself can be confusing, so I'll highlight it for you. Suggest and explain how the gliadin peptides are transported into the cells. So you have peptides. How do they enter inside cells? This is so easy.
Endocytosis.
Of course, you will write endocytosis or you will explain it a little bit.
I'll explain it. The vesicles, they fuse with the membrane, and
Very good.
they enter the cell by endocytosis.
My dear, not the vesicles fuse. The gliadin peptides will fuse with the cell membrane, and the cell membrane will engulf
Yes.
it or put it in a vesicle. Just be precise, okay? And in a question like this, you can draw, by the way. If this is the gliadin peptide, let's call it G, it will invaginate, like the cell membrane will engulf it, like phagocytosis, and then it will create a vesicle with the gliadin inside it. So it will fuse with the cell membrane, and then it will be enclosed inside a vesicle. A small drawing will be perfect. Describe the sequence of events that results in the immune system producing anti-gliadin antibodies. Now, again, I think we all noticed that this question is in every single exam, in every single variant. The steps that happen in your body after any foreign body enters, either primary vaccination dose or infection. And the question is 4 marks, 5 marks, 6 marks, whatever. Who will tell me the sequence again for the 10th time? I will not say anything. Kareem, I want you to tell me what are the sequence of events that happen in your body when there is an infection, when there is a foreign antigen. We say it nearly 2 times per week now, in every exam, in every online session. Kareem?
Can you hear me, Miss?
Yes, I can hear you, my dear.
Okay, phagocytosis will start. They will engulf
Yes.
the.
Okay, so we have the antigen, it will enter, which is in this case the gliadin. Huh? And then we have phagocytosis, correct? And then what? Neutrophils and the monocytes. Excuse me? Can someone help me?
Miss, the non-self antigen will be it will be present, and it will be recognized. The cell will be antigen presenting cell.
Yes, but you did not say it nicely. Who is going to antigen? Who is going to present it? Who will be the antigen presenting cell? Which? Huh?
Macrophage.
Yes. Okay. The monocytes or the macrophages, they will do incomplete digestion, and the antigen will be presented on their membranes, and they will be converted into an antigen presenting cell. Now, this antigen presenting cell, what does it activate? What does it do?
It activates the T lymphocytes.
Which one?
So,
To be specific.
helper. To help.
Yes. Okay. I told you an easy way to remember it. The antigen presenting cell will ask for help. I want help.
Yes.
I want help. So it will activate the T helper. And then the T helper will produce what?
Cytokines.
Yes. And then the cytokines will activate what? They activate 3 things. They activate the whole immune system.
Yes. They will activate beta B lymphocytes to T lymphocytes to produce more.
Yes.
يعني they will be
B
producing more.
lymphocytes, very good. Or the T lymphocytes, or one of them, not necessarily both of them. Or the T lymphocytes, or more what? More
Angry
macrophages.
macrophage. Yes.
Yes, okay. Or you can remind yourself this way that the cytokines will activate two things, the lymphocytes and the macrophages. And the lymphocytes, either the B or the T or both. Of course, it depends on the strength of the antigen. Now, let's focus on the lymphocytes. When the lymphocytes are activated, they go through three things. What are they?
Colonal selection, colonal expansion,
Yes.
and
And huh? I'm still surprised that you are still thinking. How many times we said it? When they're activated, they go through colonal selection, colonal expansion, and then differentiation. How
أيوة
many
صح.
times did we say that? Okay. Colonal selection, you write a sentence. Colonal expansion, you write a sentence. Differentiation depends on which lymphocyte will differentiate. Colonal selection. Where is Mariam? Where is Farouk? Where is Adham? Is Adham with us? Let me see. Adham? Hello? Who's Riham? No. Farouk. Tell me, Farouk. In coronal selection, what do we write?
You're finding the B lymphocyte T helper cell that has the same antigen or the same antigen receptor.
We will not mention the T helper here at all. The T helper will produce the cytokine. The cytokine will activate the
Sorry,
lymphocytes and huh? Okay. In colonal selection, it's very easy. You can start with the word selection. The body will select, okay, which lymphocyte is complementary to this antigen, which lymphocyte has a complementary receptor to the antigen. Yes, please be organized. It's written exactly like this in your study material. Go through it. So colonal selection, why did they call it selection? Because the immune system will select, okay, which lymphocyte has a complementary receptor. This is important, the word complementary receptor. It has a complementary receptor to this non-self antigen or to this foreign antigen. Clear? This is the sentence you will write in colonal selection. What will you write, Farouk, in colonal expansion?
They divide by mitosis.
Very good. We selected. The selection is done. After we select which lymphocyte has a complementary receptor, the selected lymphocyte, whether B or T, the same thing, they will start dividing by mitosis. Very good. They will divide by mitosis. Why do they divide by mitosis, my dear?
To produce genetically identical cell of the result.
Yes, so that they increase in number. We know mitosis produces genetically identical cells, but we keep on dividing here in this case to increase in number, to amplify, to create an army. Okay? Mariam, tell me what happens in differentiation? If the B cell has been selected, it will differentiate into what?
Into plasma cells and B memory cells.
Yes, into plasma cells. And actually, if you read the question carefully, the question said here to produce antibodies. So will we mention the T lymphocytes?
No.
No, we will not. Here we are talking about selecting B lymphocytes. Good, so read the question carefully. Here we were answering in general. If the T lymphocytes, Mariam, were selected, let's say in another question, they differentiate into what the T lymphocytes?
The T helper and the T cytotoxic.
Very good. And how does the T cytotoxic or the T killer cells, how do they kill cells?
They directly attack the cell and kill it.
Perfect. It's called cell-mediated. Antibodies, it's called humoral. immunity. They release antibodies that act in the blood. Antibodies remain in the blood. Antibodies, they do not enter inside cells. Okay, everyone? Again, this question is repeated. So at least we know, or we are sure of one question. Study it well, so at least you are sure of the 4 marks. Good. Now here are plants. Describe how sucrose is transported in phloem sieve tubes from photosynthesizing leaves to other parts of this plant. I also see this question highly repeated. Now, use label lines and label to identify the phloem and the xylem. Where is the xylem? Let me guide you. This is the xylem or the phloem. Why is no one answering? This is the vascular bundle. This is the xylem or the phloem. Farouk, Noor, Mariam. This is the xylem. Okay? Now, this is the xylem or the phloem, here or here.
Phloem.
This is the flow.
This is the phloem. Okay? Good. Now, describe how the sucrose is transported in the phloem sieve tubes from photosynthesizing leaves to other parts of the plant. I want to stop here because we took here the loading of sugar in the phloem and we took it in 2 parts, if you remember. First, we studied the movement of the glucose from the source into the companion cell. Remember the companion cell? Remember this part where we will use the hydrogen potassium pump and the co-transporter protein? And this involves cell wall, and this is apoplastic. Remember this first part?
Yes.
Or you don't? Yes. And then the second part is what? Who will remind me? After we load the glucose from the source to the companion cell, to the cytoplasm of the companion cell. Tell me, Noor, what happens after that?
When the can you say again?
The loading of sugar involves 2 parts. The first part is the loading of the glucose from the source, which is the leaf, into the cell wall of the companion cell and the cytoplasm of the companion cell. And then the step
Yes.
2 is what?
It will move into the phloem
Yes, correct. From the cytoplasm into the phloem.
from the cytoplasm. Yes, by
Through a what? What is this? Hmm?
plasmodesmata.
Yes. Look, Noor, I don't want you to tell me, you know what does this means? This means that you did not study well also. You studied, but not well. When you study really, really well, you're so sure of the answer. You do not hesitate. When you hesitate, this tells me that you studied, but you studied lightly. There is a difference between studying. You can study lightly and you can study heavily. When you study really well, you're sure of the answer and you do not hesitate. This is your problem, Noor. You're very smart, but you take things very lightly. Life is very nice and peaceful. Not now. I want you to worry and I want you to be under stress. Okay?
Okay.
So, why am I asking you this question? Because which part will you write? Will you write the part where the glucose is going to move from the source, from the leaf, into the cytoplasm of the companion cell, and you will discuss the hydrogen potassium pump and the co-transporter protein, or you will write the second part? Read the question carefully.
The second
Describe
part.
how because they said sucrose is transported into the phloem. So we are going to start from the second part, where the sucrose is in the cytoplasm of the companion cell, and then it will move through the plasmodesmata into the phloem. Okay, by what? What is the mode of movement? You tell me, Mariam. How does the sucrose move down from the cytoplasm of the companion cell into the phloem? Mariam, where are you, everyone? Hello? Mariam, you're there with me. Tell me. She's gone. Farouk, tell me. Did I lose connection?
لا
Hello?
I can hear you.
Okay, tell me, Noor, how will the sucrose move from the component? You just said through the plasmodesmata. Is this apoplastic or symplastic pathway?
It is apoplastic.
No, it's not the apoplastic. This is what I mean when I tell you you're studying lightly. The apoplastic pathway involves the cell wall. Are we discussing the cell wall here? Is the cell
لا
wall
SLU.
involved?
لا
We're not in the first
symplastic.
part. We did not mention the hydrogen pump and the co-transporter protein. We simply said that now you have sucrose and the cytoplasm of the companion cell. Hmm? Apoplastic or symplastic?
It's in plastic.
You're looking into your notes. You're moving. I can hear you.
لا
Symplastic.
والله I'm not
And how will it move? Active transport or diffusion?
active transport. No diffusion because of
Tell me. It
the
will move by, you said symplastic, yes. In this case, there will be energy involved or no, no, no. Yes. Okay. Now, what happens when the sucrose now moves from the cytoplasm of the companion cell into the phloem? You have here lots of sucrose. Then what?
I cannot see the screen.
Okay. Okay, now you should be able to see my screen. Now, this is taking
Yes.
too long. Can you all see my screen now? Now, this is taking too long. I am not happy because this again shows me that you are not sure of your answers. And when you're not, this means that you did not study well again for the 10th time. Now, this question is also highly repeated. Immunity and this question. Here, we want to know how the sucrose is going to move along the phloem when the sugars are moving from the source to the sink. How do they move? I'm not going to
By
explain it.
mass
I
flow
explained it so many
due
times.
to
How does
different
Yes,
hydrostatic.
thank you. Okay. I hope you did not look anywhere. It is by mass flow. But this is not enough. We have 4 marks here. Explain. Why does the
Due to different hydrostatic pressure.
Okay. Can you also explain at the source the hydrostatic pressure is high or low?
At the source the hydrostatic pressure is low وفي التيوب it's high في الفلويد.
What do you mean it's low? The hydrostatic pressure is high at the source because when the sugar will move into the phloem, there is a decrease in the water potential. And then the water is going to move from the adjacent xylem into the phloem by osmosis, increasing the hydrostatic pressure at the phloem. Can you please study, Noor? You studied, but like you studied while you're sitting on the bed having a cup of coffee next to you. Study seriously, please, and study while you're writing things down. So there is high hydrostatic pressure at the source. And you need to explain why did the hydrostatic pressure increase at the source? Because when the sugar has been loaded into the phloem, the water potential in the phloem will drop. And then the water is going to move from the xylem into the phloem by osmosis, increasing the hydrostatic pressure at the source. Now, can you tell me or discuss hydrostatic pressure at the sink, Farouk? Where is Farouk? Hydrostatic pressure at the sink is high or low?
Uh, uh, it's low, high, low,
Habibi. No, no, no. It's high or low?
slow.
It's low. Why? What is happening at the sink?
Yeah, yes.
Okay, I'll explain this
Unloading
part.
of
I'll
sucrose.
explain. Yes, I'll still explain this part. It's okay. Now we have here the source. Pay attention, everyone. We have the source, the leaf. It is producing sugars. It is producing glucose, to be specific, because there is photosynthesis. And then this glucose is going to be loaded into the phloem. This is my phloem. 1 minute. Let's choose blue. This is my phloem. And the phloem is accompanied by a companion cell. This is the companion cell. We have a two-step procedure. First, the glucose is going to move from the source into the cytoplasm of the companion cell. And how this happens, if you remember, by the hydrogen potassium pump and the co-transporter protein. You have here the cell membrane of the companion cell, and you have here the cell wall. You have here a hydrogen potassium pump, and you have here a co-transporter protein. What happens? We always start with the hydrogen pump. The hydrogen pump from the name. It will pump hydrogen ions against a concentration gradient from low concentration to high concentration. The low concentration is from the cytoplasm of the companion cell into the cell wall of the companion cell. And then you will have a high concentration of hydrogen ions in the cell wall of the companion cell. And then the co-transporter protein will work. The hydrogen ions will move down a concentration gradient by facilitated diffusion. And while the hydrogen ions are moving down a concentration gradient by facilitated diffusion from the cell wall of the companion cell into the cytoplasm of the companion cell, what will be co-transported with it? Who will remind me? At the same time, what will be co-transported with the hydrogen ions?
Sucrose.
Glucose. Glucose will be co-transported with it against a concentration gradient from low to high. And this is, of course, going to be an energy consuming process. Now, Mariam, I want you to focus with me. The glucose is going to be co-transported, not the sucrose. When the glucose enters the cytoplasm of the companion cell, inside the cytoplasm of the companion cell, the glucose is converted to sucrose by a particular enzyme that you did not study. Okay, Mariam?
Okay.
Okay, my dear. So the glucose will be produced in the leaves by photosynthesis. And then the glucose will be transported into the cytoplasm of the companion cell by the method that we took, by the hydrogen potassium pump and the co-transporter protein. The glucose is now inside the cytoplasm of the companion cell. The glucose is converted into sucrose first, and then the sucrose is going to move through the plasmodesmata by diffusion down a concentration gradient, symplastic pathway, so that the sucrose now is found in the phloem. This is the loading of the sugar into the phloem. It is a two-step procedure. First, we will discuss what is happening at the cell wall of the companion cell so that the glucose moves from the source to the cytoplasm of the companion cell. Inside the cytoplasm of the companion cell, the glucose is converted to sucrose, but this part is not very important. But Mariam noticed this because she's smart. And then the sucrose is then going to move from the cytoplasm of the companion cell across the plasmodesmata into the phloem. Are you all with me? This is the first part. Now, the second part, the sucrose will move from the source to the sink. How does it move? By mass flow. Noor said it, by mass flow. Mass flow is a general term, by the way. It is not only confined to plants. It is confined to the movement of any fluid from high pressure to low pressure. Also, mass flow, we mentioned it when we were discussing the movement of blood in blood vessels and arteries, in veins. Always fluids in this situation, in mass flow, moves from high pressure to low pressure. In the case of plants, we're talking about high hydrostatic pressure. Okay, do you all understand what I'm saying? Does anyone want me to repeat anything? I did not finish, but up to now, is it clear?
Yes.
Good. You have high sucrose in the phloem at the source. So this means the phloem is concentrated. There is little water potential. I have initially little water. So when I have low water potential in the phloem at the source, this is going to attract water from the nearby what? What do I have next to the phloem?
Xylem.
Exactly. The xylem is going to give the phloem water. Water is going to move out of the xylem by osmosis, and now the phloem will have lots of water. Initially, it was concentrated when it was filled with sucrose and it had low water potential. Because of this low water potential, it is going to attract water from the nearby xylem. Water is going to move out of the xylem by osmosis and it's going to enter the phloem. So the phloem now will have high hydrostatic pressure. It had low water potential initially, but after the water moved out of the xylem into the phloem, it will have high hydrostatic pressure. So this high hydrostatic pressure will push the flow or will push the phloem sap from the source and it will go where? To the hmm?
Sink.
From the to the sink. At the sink, I am unloading sugar. Sugar is going out. Sugar is going out. Sugar is going out. The sucrose is going out and it is being stored in the fruits. So at the sink, I have high water potential or low water potential.
High water
High.
High water potential. Okay, so I have lots of water in the sink because the sugars are going out. This high water potential or lots of water, water will always move from high to low concentration. So the water is going to move from the sink back to the xylem by osmosis down a concentration gradient. And when the water moves out, what will happen to the hydrostatic pressure at the sink? It will be high or low?
Low.
Low. The part that is confusing is the hydrostatic pressure and the water pressure. I advise you to ignore the water pressure part, the initial change in water pressure, because it confuses you. You just simply need to know really, really well that first step in the transport of glucose from the source into the companion cell using the hydrogen pump and the co-transporter protein. I hope this is clear. And then the second part, which is the movement of the sucrose from the cytoplasm of the companion cell into the phloem through the plasmodesmata. When the sucrose enters the phloem, you can say that the phloem will become concentrated. Don't mention water potential if it confuses you. So the phloem will become concentrated. When it is concentrated, it will draw water out of the xylem and the water will move by osmosis down a concentration gradient. So at the source, you'll have high hydrostatic pressure. At the sink, the sucrose is unloaded. So the sink will now be less concentrated. It will be dilute because it is unloading. Because it is dilute, the water will move out. So at the sink, you'll have low hydrostatic pressure. And the changes in hydrostatic pressure will move the phloem sap from the source to the sink by mass flow, mass flow, mass flow. Mass flow is the flow of fluids from high pressure to low pressure. In plants, the pressure here is hydrostatic. Okay? And mass flow also happens in blood vessels. The blood always moves from high pressure to low pressure. Now, does anyone have any questions over this matter? Is it clear?
No.
Yes, clear.
Okay, very good. Now this exam doesn't finish. Suggest why the treatment of photosynthesizing leaves with Cn minus results in less sucrose being transported into phloem sieve tubes. Now we have to read the question. This cyanide ions inhibits the activity of an enzyme. So now it's clear why when we treat the photosynthesizing leaves with cyanide ions, there is less sucrose being transported. It inhibits the activity of the enzyme. So what is it? Hello? Cyanide
Yes,
ions inhibit the activity of an enzyme involved in respiration. So it prevents respiration. When we treat photosynthesizing leaves with this enzyme, there is less sucrose being transported. When it is affecting respiration, then what it is affecting the production of what?
ATP?
Yes. Again, this is an easy question, but it is misleading. When respiration is affected, this means less or more ATP.
Less.
Less ATP.
Very good. Less ATP, less energy, and definitely sucrose transport is an energy consuming process or not an energy consuming process.
Energy consuming.
Yes, so when I have less energy, I will not be able to transport the sucrose. Don't forget the action of the hydrogen pump and the co-transporter protein. Those need energy. So I will have less energy. Okay, explain your answer. No, I have to know. A student carried out a semi-quantitative Benedict test on two solutions. Solution A was extracted from the cytoplasm of cells in the mesophyll tissue of photosynthesizing leaves, and solution B was extracted from the phloem sac. The solutions were taken from the same plant. This is tricky. Look here. Solution A came from the leaves and solution B came from the phloem. Which one will have glucose and which one will have sucrose? The leaves will contain glucose or sucrose?
Glucose.
Yes. And the phloem will contain glucose or sucrose?
Sucrose.
Good. Perfect. Do you all understand why? We of course, we will
Yes.
know well that glucose is converted to sucrose for translocation because it is less reactive. So the Benedict will give me a positive result in solution A or B.
A.
Excellent. Bravo. The solutions were taken from the same plant and the other variables were standardized. For each solution, the student measured the time taken for the first color to change. So suggest which of the solutions, A or B, would show the change in color in the shortest time. Explain your answer. Mariam said A. Benedict will give me a positive result in A, and the color change will be positive in the shortest time. In B,
It will not show a positive result with Benedict because it's a non-reducing sugar.
excellent. It's a non-reducing sugar. Okay. Or you can say in B, it will take longer time because there might be traces of glucose. But what you said is correct. It's non-reducing and it does not release or donate electrons. It will not reduce the Benedict. So the Benedict will not show positive results. Very good. You have to say, of course, here you have glucose, here you have sucrose. So this also is an easy but tricky question. They're all easy actually, but how they write it makes it tricky. The last question. Polysaccharides such as glycogen are composed of thousands of monomers. Oligosaccharides are carbohydrates that contain 3 to 10 monomers in their chain. Nistose is one example of an oligosaccharide, and the structure of nistose is shown in figure 6.1. Okay, state 3 differences between the structures of the nistose and glycogen other than the number of monomers. Now, this is a question that depends on your studying of biological molecules. Can you tell me glycogen is made up of what?
Alpha glucose.
Alpha glucose. Okay, look at nistose. Look closely at the molecular structures. Are they the same? What
No,
is this?
it's similar to a
Glycogen
fructose.
is branched.
Excellent. This is one difference. Good. Number one, this is a very obvious difference that the glycogen is branched. Correct. But we want two more. Now, the glycogen. What else do you know about the glycogen, Noor, that is not present here?
I know that the glycogen has 1,4 and 1,6
Very
glycosidic
good.
bonds.
Very good. Now, the linkages here in the glycogen are 1 to 4 and branching at 1 to 6. Here, the linkages, I don't expect you to really know it, but are they 1 to 4 and 1 to 6? This is
No.
actually 1 to 2, but I will not expect you to know it. Okay? No. So you can mention the linkages that you know, 1 to 4 and 1 to 6 in the glycogen, and you can say that in the nistose, they are not the same. And this will be enough. Okay? They're
But
not the same. So you don't have, yes,
also the nistose have more than one type of monomer.
yes, this is what I was trying to get you. But Noor, he jumped to the branching and the non-branching part. Of course, it's very obvious. This is an alpha glucose. What is this? You know it, by the way. But it is inverted. Who will, like, this is what? It's in the sucrose.
Fructose.
This is the, yes, this is the fructose. If you remember the structure of the fructose, you have to know it. The fructose, CH2OH, CH2OH, OH, OH, and then OH up here. Okay, so this, if you don't remember, you can simply say that the monomers are not the same type. They're not the same number. They're not the same type. Okay, the glycogen, write what you know. You can say that the glycogen, all of them are alpha glucose. But in the nistose, not all of them are alpha glucose. They're different. Just like Mariam said, they will give you the grade. And if you're sure what they are, you can mention that the nistose is glucose. And if you noticed that this is the beta fructose, then write it down. Okay? So this question is also tricky if you did not study biological molecules well. So we will write here, not the same monomers, not the same type of monomers. In glycogen, they're all alpha glucose. In the nistose here, you have fructose and alpha glucose. Glycogen, only alpha glucose. Describe the roles of the rough endoplasmic reticulum and the Golgi body in synthesizing glycoproteins. Now, this is too easy. This is an O-level question. What is the function of the rough endoplasmic reticulum?
Translation.
Yes. The rough endoplasmic reticulum translation. What else?
Modification of proteins.
Very good, modification. Give me examples of modification.
Removing the first amino acid.
Yes, removing the first amino acid. What else? How do we
Adding,
change it to a glycoprotein?
adding lipids. Carbohydrate.
Yes, adding carbohydrates, adding carbs. Good. Now, can you give me one function for the Golgi body? Two functions.
Modification
So
and
2 and 2. Yes, modification and packaging. Very good. Packaging to go where?
either turn into a vesicle that is used inside the cell or leaving the cell by exocytosis.
Very good. Very good. Also, the rough endoplasmic reticulum packages and transport vesicles so that it goes to the Golgi body. So this is easy. State one role of the glycoprotein in the cell surface membrane. Now, this is easy also.
Acts as a
Cell
ligand.
signaling.
Excuse me, I did not hear you. You both talked at the same time. A glycoprotein on the cell surface membrane, what does it do?
Cell signaling.
Yes, cell signaling, a molecular antigen, cell-to-cell adhesion, okay, a molecular marker, anything, a receptor. Fine. Oh, we finished the exam. Thank God. Now, this is an easy exam, but I tell you, easy exams are, they have a very high curve. Look, please, 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 I want you to do really well in paper 2. 50% of the grade goes to paper 2. Paper 1 is a deceptive paper. It is a very deceiving paper. Like paper 1, there are 40 questions. 20 questions are easy, and the other 20 questions are very tricky. They are made tricky. They are intentionally tricky. So no one aces paper 1, but you can, of course, do well. The more you practice paper 1, the more you will do well in paper 1. And we have lots of time before paper 1. We will solve a lot of paper 1. But please, I want you to focus a lot on paper 2. Paper 2 is most of the grades, and paper 2 is easy if you study well. So this exam was very easy. Okay, I already saved it. This was a question that was tricky. I highlighted that for you. The immunity question is repeated for the 10th time. And the loading of sugar is also a very common question. And I highlighted those for you that needed just give it a second look. Okay? And I can see that you are all improving. You're studying more, but I still want more studying. And please study with concentration. Don't study lightly. Don't study while you're drinking a cup of coffee and you're sitting on the couch. Study with concentration and write down the answer as if you are writing it on a real exam. Okay? Any questions?
No, thank you.
No.
Okay, today is what, Monday or Wednesday? I'm losing track.
Today's
Monday.
Monday.
Monday. Okay, you all have paper 3 on the 28th. No one has paper 3 on the 9th. So on Wednesday, we will meet online for another exam. And next week, we will go to the lab on Monday, not Wednesday. We're not in a hurry. Clear?
So next Monday, it's physical.
Yes. No, next Wednesday, after 2 days. Tomorrow is Tuesday. And then we have Wednesday.
Okay. So this Wednesday, after tomorrow is it's just past papers, and then the Monday
What
after
do you
that.
want? You want to go to the lab or you want to do past papers? We're not in a hurry. No one has exams on the 9th. You all
Yeah.
have exams at the end of May, after a month. So next week and the week after, we can go to the lab.
I don't mind either.
Okay. So I
This
think that on Wednesday now.
so this يعني
Let's
week we will have past papers بعد كده after that lab.
عشان your exam is next month, not on the 9th. I panicked because I thought that one of you had an exam on the 9th. No one, none of you.
Yes.
I checked from the office. So all of you, your exam is on the 28th. You have more than a month. Okay. And we need to go to the lab twice maximum. You will remember everything. Okay. Actually, we can also do paper 3 online together, like we used to do in class. But on Wednesday, we will meet online and we will solve. I'll decide. Let's solve paper 3 online. We'll think. But on Wednesday, it will be online. Next Monday and next Wednesday, it will be in class. It will be in the lab, I mean. Okay?
Okay.
Okay. So see you Wednesday. This Wednesday online. We'll solve another paper too. Good luck.